Okay, we published a new scientific paper, and we in this case are my friends and colleagues Bart Ermentrout and Joe Lanzittel. And this paper contains what we believe is the first ever simulation of a Goby brain. Now, gobies are, of course, small, often cryptobenthic fishes, marine and freshwater fishes, and I've been thinking about gobies and investigating them in the ocean for about 10 years, and I've been thinking about brains for about 20 years. So it's lovely that these two strains of intellectual inquiry came together. Now, what is the paper about? About 120 species of gobies are so-called shrimp gobies, meaning they live in a symbiosis with an alpha-8 shrimp. These alpha-8 shrimps dig burrows. In these burrows, the gobies and the shrimp live together. Now, what's the benefit for the shrimp? The shrimp gets a watchman. So the goby perches at the entrance of this burrow, and it warns the shrimp of oncoming danger. So if there is a full-blown threat, like a predatory fish approaching fast, the goby will escape into the burrow just like any regular fish escape, and the shrimp will also remain underground. Now, there's an intermediate level threat signal, which is the so-called tail flick reflex. And in this case, the goby flicks either its tail or its dorsal or its caudal fin. And the shrimp, when it's above the surface, it's constantly in touch with the goby, and it senses this tail flick. So the big question we try to answer is, how can this come about? What is potentially the neurobiology of this tail flick reflex? Now, what we did, we used computer simulations. We started with simulations which were designed to describe the zebrafish escape reflex. So these simulations had a central pattern generator which generates tonic swimming, as well as a circuit which receives a threat and conveys the information to this threat to these motor neurons, where then a escape reflex starts to happen. Now, what we did with our computer simulations, we speculated. We asked, how do we have to modify this space model in order to get it to reproduce not just a full-blown escape, but also this tail flick intermediate signal from the goby to the shrimp? Now, what we found, two modifications were necessary to create this. And one was that we shut down the activity of the central pattern generator for swimming in a base condition, which is realistic because usually the gobies don't swim. They perch in front of this burrow, they sit still. The other thing which we did is we introduced a direct connection from the senses perceiving the threat, and this would be something like a visual perception or an auditory or a lateral line perception. These senses then are connected directly to the central pattern generator, and then that caused a tail flick reflex. This is not present in conventional fish, non-shrimp gobies, so this extra connection is what gives rise to this unique communication system in the shrimp goby. We believe this is a realistic proposal. There are neurons in the brainstem which are homologous to these famous Martner cells. They are essentially in the same region as the Martner cells, just a little bit smaller. These neurons could lead to an additional indirect pathway from the threat perception to the central pattern generator involved in moving the tail or the fins of the fish. So I think this is an interesting study which uses theoretical neuroscience to propose how a very curious evolutionary innovation, this new communication system in the shrimp goby came about. The paper is down in the links in the description. Also the shrimp goby page, which is a page I made which describes the symbiosis between the shrimps and the shrimp gobies in detail. It's also linked just below here. I hope you found this interesting. Please don't hesitate to contact me if you have any questions regarding this study, and see you next time.